Start recording also. Alhamdulillah. Alhamdulillah. My screen is visible to all.
Yes, ma'am.
Yes,
Okay.
ma'am.
So now here we will be discussing paper 1 as well as paper 2. Now you all know that in case of paper 1, there is nothing much to discuss. Like whatever we have studied, that only we have to write in the paper. Basically, paper 1 is a bit like you are sitting writing lot. Okay. So now when we talk about the paper 1, so paper 1 may whatever is given in your handbook, everything you have to buy. It is expected from the examiner. Okay. Now, question number 1 in paper 1 is for 9 marks. That is sepia, sketch and label, digestive system or reproductive system or nervous system. Okay. Now, after that, there is another option. Now, see, if this particular question has been asked, sketch and label the system. So, in that case, it is expected that you will draw a very neat and clean diagram. First thing. Second thing, the diagram should be properly labeled. Third thing, you should know about the system. Is that clear? Now, in that, you will have an option of describe A, B, C. Now, in that, jaws, radula, chromatophores, from spermatophore, statocyst, all this will come. So, any three option will come. Okay. So, you have to identify and describe. In that case, the diagrams will be given. You have to identify that particular diagram. And student, you can see the another slide now. Can you see the slide of the question paper now?
No, ma'am.
No,
Ma'am. It's only first slide.
Yeah, that's what I wanted to confirm because this happened in some time that I have observed that first slide is only shared and this is very new for me. So that's why I wanted to confirm now. Now?
No, ma'am. No, ma'am.
No, ma'am.
Still you cannot see the question paper?
Now it is visible.
Now
Question
it
paper is
is.
visible, children?
Yes, ma'am.
Okay. Actually, this is my also new experience to use this Zoom. Earlier, I was very much used to with the Google Meet, but I could not connect with that. Let it be. Now come to the point. Now for question number 1, I hope first option everyone is clear, right? Sketch and label the system. Any doubt in that? Any doubt in that?
ma'am.
So
No,
you
ma'am.
have to draw any
Ma'am,
of the
like
system.
we have to choose from these
No,
three
no,
or
no. Absolutely no. Sorry. This will be the examiner's choice. I told you in examiner's point of view, it is expected that TYBSc students sitting for the examination should have the thorough knowledge of everything which is given in their handbook. Actually, basically the syllabus. Okay. Now this skeleton paper is given in your handbook, page number 67. Okay. So sketch and label may you will be getting any of the system for the 9 marks. I told you what is the expectation of the examiner, that you will draw a very neat and labeled diagram. You know about that particular system, or you will be given any 3 diagram of jaw, radula, chromatophore, spermatophore, statocyst, and you will be asked to identify and describe. Now, wherever identify and describe type questions are there, na bacha? So in that case, it is expected if you will identify correctly, you will be getting 1 marks and 2 marks are for the description. Very well. Question number 2 पे आ जाते हैं. Identify and classify giving reason. I hope you all remember when we were doing the regular practical. So that time we have done this particular thing. Identify and classify giving reason. Now same way it is given in your handbook. If the phylum is protozoa, why it is protozoa? You have to write, and it is already given in your textbook in a single line. So here also in A, you will be getting 3 option: protozoa, porifera, or Nidaria. Means out of these 3, only one specimen diagram will be given, or the specimen. Basically, if it is a PowerPoint presentation, they will be giving you the diagram, or they will be keeping the specimen. Okay, now in B, platyhelminths or nematoda. In C, annelida, arthropoda. D, mollusca or the echinodermata. Now everywhere this classification is already given in your handbook, in that the reasons are also mentioned. Question number 3 पे आ जाते हैं बच्चों. In question number 3, basically if you will see, you will be having Acanthocephala, then it is a Chetognatha and Onychophora. This is minor phyla if you remember. And option B is Hemichordata. Option C is important here, please pay attention. Observe the animal. Now it could be any photo, it could be any preserved specimen. Now after that, here basic thing you need to identify, that is the phylum. Say example, if I have kept one cockroach, okay, I'm just giving you basic example, don't expect the cockroach in the examination. Any invertebrate will be kept. It could be the earth, other than syllabus bola, earthworm is given in your syllabus. So example, I think cockroach is not given. So say example, cockroach is kept, housefly is kept. Okay. Basically, you should be able to identify where arthropoda hai. Is that clear? It is expectation. And after that, you have to write the description. Now, how to write the description? Now you understood arthropoda hai, this is insecta. The simple way, whatever description of that particular class is there, that you have to write here. Question number 4. Field report, submission and discussion based on any field visit which is attended by the student to avoid disparity due to the financial constraint. Now see, we all know in our syllabus, basically we have the 3 field visit, right? So any field visit is okay. So we have gone for the 3 field visit, right? So out of 3, you can write any one. Any one is expected to be there in your syllabus, in your field book. So field book is must. Now if you have made the field book, please come with the field book. Don't forget your field book, as you need to remember that you have to have 10 marks. Now see, submission and discussion. So here also it depends on the examiner. Basically it is divided in 6 and 4, means writing the report, submission, and if you have made a proper field book, so you will be getting 6 marks and 4 marks on the viva, whatever. Now next question पे आ जाते हैं, that is the question number 5, viva was. बच्चों, this question, this viva will be based on the theory, all right? And your journal, now see, you have a very well printed journal, so you're expected that it is properly covered, it is properly certified, and the certified means the your class teacher and your head of the department signature is there on that whoever mic is on, I request to mute it. Whoever mic is on, I request to mute it. Okay, shall we move ahead? Now,
Yes,
next slide is visible to you, students. Question paper 2.
No, ma'am.
No, ma'am.
I think for changing every slide, I need to go for a new share or what? I don't understand. Just a minute. Let me see. How can I share the entire PowerPoint presentation? Okay. Now it is visible. Now it is visible.
No, ma'am.
I don't understand.
No,
Every time I need to change the PowerPoint presentation. If I'm changing a slide, it is asking me to change this. Now it is visible. Question paper 2.
Yes, ma'am. Yes, ma'am.
Now, paper,
Yes, ma'am.
now students, paper 2 is not like to discuss entirely like I have discussed into the one slide. I have separated it. Now, question number 1 is visible on my screen. Question number
Yes, ma'am.
yes or no? Can you see
Yes,
the
ma'am.
question
Yes,
number
ma'am.
1 slide only?
Ma'am.
Oh, Allah Tera Shukr hai. Good. Enumerate the erythrocyte in the given sample and comment on the clinical condition. Now, see, question number 1 is for 15 marks. We have 3 options. Now, enumerate erythrocyte in the given sample and comment on the clinical condition, or enumerate the leukocyte in the given sample and comment on its clinical condition. Now, I kept it in a single slide because it has a same type of assessment pattern. Now, when you are doing the dilution and charging, now we all know the process that whenever you are performing this particular practical, so that particular time, first of all, you are cleaning your RBC or WBC pipette. After that, you are going to take the blood in that, dilution liquid in that, and after that you are just rotating it between your palms, and after that you are discarding 2-3 drops, initial drops, and you will be charging your this neoburst chambers. So till here, for this particular process, you will be getting 4 marks. After that, you have to observe that particular slide in the microscope. Basically, bachcho, you should observe it under the 10x and after the 40x and start enumerating. So when you will count, now see, there will be multiple microscope. So you have to just count one microscope, and you all know how to draw it in your book, right? You have already drawn it. So for that, you will be getting 4 marks. Calculation part contains the 4 marks. Explanation contains the 3 marks. Okay. So this way the marks have been distributed. See, it depends on the examiner also. They may change the marking scheme, but basically my idea is that marking scheme will be like this only. Now, presentation or report on differential count of leukocyte and comment on clinical condition. I hope same is appearing on my screen. Is it?
Yes,
Yes,
ma'am.
ma'am.
All right. So when we talk about the differential count of the leukocyte, now you all know you have to take a drop of blood, then staining. A long process is there that you know very well. So for making the smear, you will be having 3 marks, staining 4 marks, identification of at least 2 type of cell. Now, see here what you are identifying. That particular cell, examiner should also be able to see. This thing is very much important. Result and explanation. Now, see, student, for this particular thing, examiner will come to your desk and will take the table viva. All right, or they may call you on their own desk. It depends on the examiner to examine. Now, let us go on the question number 2. Question number 2 दिख रहा है screen पे?
Yes, ma'am.
Now I am sure. Now I will not ask repeatedly. Now question number 2, estimate total plasma protein by Follin's method, or estimate serum plasma total triglyceride by phosphovanillin method. Now student, both the questions are for the 10 marks. इसमें से कोई एक option आएगा. Okay. Now chemical reagent. ये केमिकल और रिएजेंट के बीच में बाय मिस्टेकली जो है कॉमा आया है. केमिकल रिएजेंट जो कुछ भी है, that is expected that you should know before performing. Now see, in when I was explaining you this particular practical in the class lecture, that time also I had stress on this thing, that you must know before starting your procedure. You should know what are the chemical reagents required to you, what is the principle, and if any reaction mechanism is there, you should know. And once you have done, then only the examiner will allow you to perform this particular practical. So for that, you will be having 3 marks. Now your experimentation, your observation table, formula, calculation have the 5 marks. Formula is very simple. Now you all know it. It is OD of unknown divided by OD of standard into the concentration of the standard, as well as if you have any dilution factor or conversion factor, that will be there. Now, see, it is expected what are the normal value of the protein and what is the normal value of the triglyceride. It should be known to the student, and what are the clinical significance of that, and what are the diagnostic method should be known by you. Now, how to perform this particular practical? Now, student, I hope you all have your handbooks in your hand and you are watching the same in your handbook as well. So you will be basically preparing 3 test tubes. Now see, these are your 3 test tubes. These 3 lines are your 3 test tubes. So standard, unknown, blank. In standard, you will be pouring 1 ml of BSA solution and 5 ml of alkaline reagent, alkaline C reagent. After that, second test tube, unknown, 1 ml this serum. And alkaline reagent 5 ml. Blank में 1 ml distilled water, 5 ml of alkaline reagent. After that, you will be waiting for 15 minutes. Stand there only for 15 minutes. Then after that, you will be adding the folinsilic alkyl reagent in all 3 test tubes and mix well, keep it dark for 30 minutes. You have to take the OD of it. Now next, practical estimation of plasma total triglyceride by phosphovanillin reagent. Here also you will be preparing 3 test tubes, students. Now 1 ml of distilled water and 2 ml of concentrated H2SO4 sulfuric acid in blank test tube, 1 ml of standard lipid and 2 ml of concentrated H2SO4 in your standard, 1 ml of serum or plasma and 2 ml of concentrated H2SO4 in 1 test tube. So this 3 test tube you keep for the boiling water bath for 10 minutes so that the digestion of that particular lipid will happen with the acid. Now, examiner may ask you question why you are keeping it in water bath. So, in the hot temperature, this lipid will react with the acid, and after 10 minutes, you will be taking it out and you will cool that particular mixture, and after that you will be using it for the step 2. Now in step 2, only 0.5 ml mixture you will be taking. Now student, step 2 estimation, you should be very careful. Okay, when you will be taking the mixture from step 1, so in step 1 mixture you should remember that the mixture is made up of concentrated sulfuric acid. So you should be very careful while taking it. Plus, you will be adding the phosphovanillin reagent in which the concentrated orthophosphoric acid is there. So you should be very careful while doing this particular thing. Now, question number 3, I think I need to stop for a minute. Then I will start question 3.